I run a nonprofit called Project Insulin. We're making our own biosimilar insulin glargine, and we're going to sell it directly to patients at cost, regardless of insurance status. Our mission is to ensure that everyone who depends on insulin has access to it. When I started it, the main concept was making insulin affordable is not a scientific problem, it's a money problem. People know how to make insulin. It's one of the first molecules people work with when they're in undergrad or a master's or a PhD in the sciences. So it's not like finding someone who can do the drug development work would be that hard. Really, the issue is fundraising. Can we raise enough money to actually make an insulin that is affordable and available to anyone who needs it?
1 in 5 insulin users in the US rations their supply. A full century after the molecule was discovered. Why is something this essential still out of reach for so many? Today, Eric Moyal, founder of Project Insulin, joins us to take that question apart. We trace his path from nonprofit fundraising into biotech and start unpacking what it actually takes to build an affordable biosimilar insulin. Let's get into it. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Eric. It's good to have you on today.
Thank you. Great to be here. Thanks, David.
To start us out, let's start with this controversial question. Share something that you believe about bringing medicines to patients that most people disagree with.
Interesting. So my first thought would have been that everyone should have access and it should be affordable. But I think that actually most people do agree with that. And in fact, many people I meet in the biotech space firmly believe that. So I would say my hot take, so to speak, would be a nonprofit could make life-saving medications.
Interesting. And we're going to talk more about that. I'm very excited how that could play out. How did you get actually attracted to science? What was this spark? What drew you into the world of drug development? And what were the pit stops along the way to now developing insulin?
It's a long road filled with twists and turns. I am not a scientist. I don't have background in any of the scientific fields, biochem, neuroscience, anything like that. I went to university, I got a business degree, ended up in university fundraising, and ended up in life sciences because I was looking for a way to make a change in healthcare. Well, the opportunity to do so appeared in the form of creating a generic drug, using my skills as a fundraiser to fund a project and then hopefully use the skills of scientists, hire people to actually do the drug development work itself.
What intrigued me is when I came across your endeavor to develop insulin, and the first thought that crossed my mind, wait a minute, insulin has been around for more than a century, but yet there's still an affordability crisis, in particular in the US. Am I missing something? What is the problem there? Tell us.
It's interesting. And insulin was originally discovered over 100 years ago, and the original patent was sold for $1. And the researchers famously said, insulin does not belong to me, it belongs to the world. And so how do you get from a place started off with such good intentions to a place where now it is so unaffordable for so many people and people are rationing insulin? How we got here, I'm not sure. As far as the full details, I couldn't tell you. I'm not an anthropologist, but what I can tell you is that the most recent versions of insulin, another word for synthetic analog. So the most recent versions of insulin, the analog insulins, the patents for those have expired. Those were invented in the late 90s. And there's a few things to blame for the high prices. There's investors. Businesses have a fiduciary duty to their investors. So that means that anyone who is making a drug literally has to make as much money as they can because that is the deal that they make with their investors. They have a fiduciary duty to those people. Inherently, the model is focused on profit. A secondary focus is always people's health, and some of the major pharmaceutical brands include people's health as some of their main goals in their financial statements every year. But again, it has to be secondary. And so there's that piece. And then I think in the US, we do not have a single payer system. It's not government-based healthcare. It's privatized. And as a result, there's quite a lot of middlemen organizations that are part of the insulin distribution process. So we already talked about manufacturers, but there are wholesalers, insurance companies, the pharmacies, and a few other middlemen organizations that all need to make a profit on the journey of getting insulin from a manufacturer to a patient. And so each of those extra steps adds to the cost. And so a confluence of issues leading to high prices, but for-profit shareholder-based companies and convoluted distribution systems, I would say, would be the two major pieces.
And today, how many patients in the US can't afford insulin, and how big is that?
So the statistic that's out there is 1.3 million Americans ration their monthly insulin supply. So that's 1.3 million Americans who don't use as much insulin as they're prescribed to be using in a month. Part of that is due to how expensive it is. For others, it's not having access, but a large part is how expensive. The current statistic is because the landscape continues to change. But on top of that, it doesn't include the people who are buying their insulin, but are making other sacrifices. They're not eating healthy, or they don't have the means to get a better education or quit a job because they have good insurance at their current job, but they hate the job. There are other things that count towards, I guess, like inaffordability or access that aren't necessarily just, I literally can't pay for this. So the number is 1.3, but I think a much larger group also includes that portion. And unfortunately, I've gotten emails. I mean, we've been doing this for 5 years now, and every couple of months I'll get an email or two saying, hey, my insurance, I can't afford it this month, or I got laid off and now I don't have insurance, or I'm 27, so I'm off of my parents' insurance now, which is a major issue here. There's like a whole lot of stories that I hear all the time. And if you go to our social media, our most recent posts, a lot of people have commented about how they've struggled.
1.3 million, help us to understand how much that is in perspective of the total patients. What is the percentage there?
So it's 8 million patients who are insulin dependent. So not everyone with diabetes uses insulin, but of the 8 million who are insulin dependent, it's 1.3. So it's about 20%.
That's a big number, definitely. So tell us, since you have a unique background, you're not coming from the biological or biotechnology side, how did your work from fundraising and nonprofit leadership actually influence the way you develop now a biosimilar insulin?
Well, first, there is something to be said about coming from a different field, not having the preconceptions and notions of what can and can't be done in life sciences or pharmaceuticals is a strength. There's a lot of weaknesses. Like, I didn't know anything about anything. So that took time to learn, but there is value there. The value is no preconceived notions. So I can why can't it be done this way? So I run a nonprofit called Project Insulin. We're making our own biosimilar insulin glargine, and we're going to sell it directly to patients at cost, regardless of insurance status. Our mission is to ensure that everyone who depends on insulin has access to it. That would be the soundbite. When I started it, the main concept was making insulin affordable is not a scientific problem, it's a money problem. People know how to make insulin. It's one of the first molecules people work with when they're in undergrad or a master's or a PhD in the sciences. So it's not like finding someone who can do the drug development work would be that hard. Really, the issue is fundraising. Can we raise enough money to actually make an insulin that is affordable and available to anyone who needs it? So that was the original thesis. Of course, over time, I've learned how scientifically challenging it is and all these other pieces. But that main idea that if we can raise money, we can tackle this problem has been consistent. Even if it is more complicated to make insulin than I thought, if you have the right people, it's still more than feasible. I'd say that fundraising is really the driver of this whole thing. That the idea that if I can raise the money, we can actually make an insulin and get it to patients has driven and continues to drive us forward. So I think the background in fundraising is incredibly important and is the only reason why I even thought I could tackle this, because I'd wanted to be in healthcare previously or realized that I really am passionate about healthcare on a more general level. But a lot of things require research, and I'm not a scientist, I'm not a researcher. So finding an opportunity like insulin really was exciting for me because I could use my skills to do something that would impact a lot of lives in healthcare.
Raising funds is challenging, no doubt about that, especially nowadays. Besides that, the technical development, you alluded to that, is also challenging, even though insulin has been around for quite some time. And as you well know, smart biotech scientists, developing a biosimilar is a challenge. Can you tell us a bit how you go about on the technical side, from the strain and process development through the analytical? What is your strategy there?
My strategy was hire someone really good. So I will divulge some details, but I can't give you everything. But we hired a chief scientific officer once we were far enough along in the fundraising process that we could offer someone a respectable salary. And she has 18 years of experience working at Merck, specifically the biosimilar insulin we're working on. And so I found the best person for the job and I got really lucky. And so hiring her was a huge boon for, well, the technical side, which had previously been a few advisors kind of leading me on the path and giving me a 20-minute YouTube degree in in the life sciences. So the way we're building it is that we're hiring a CDMO to actually do the drug development work. We're not having our own lab space and kind of building it that way. We're working with a CDMO. When it comes to that, we need to ensure that we have the right metrics that we're asking for when it comes to the development process. And so given our chief scientific officer's background, her name is Allison, so I'm just going to refer to her as Allison. But given Allison's background, I feel very strongly that she'll be able to oversee it well. But to answer your question on markers, titer is important. How pure we can make it is important. And I know you have a more technical viewership, so I apologize in advance for simplifying things. But this is how I've learned it. I've learned it through people who know it well. But just ensuring that it's pure and we can get as much titer out of the molecule as possible will downstream lead to more insulin per bioreactor, which brings down our costs, et cetera, et cetera. So those would be the bigger pieces that we're looking for when it comes to the variables. But additionally, we picked a CDMO who's got a really strong analytical track record. And that was on purpose. We wanted to ensure that given the FDA's, not latest, but recent-ish moved from biosimilars to be considered more like a generic, which wasn't the case before in the US. Obviously, in Europe, it was different. The goal is interchangeability. It's showing biosimilarity and then eventually interchangeability with the original product, the reference product. And so ensuring that we have strong analytical data is a huge part of submitting to the FDA. A, hire the right chief scientific officer. Huge step, obviously, to making a sound product. And then B, looking at that 30,000 foot view of like, okay, well, I need someone to make the drug. Anyone can. Well, anyone with the correct bioreactors and correct equipment can. But thinking ahead of like, okay, well, it's not just making it, it's showing a robust analytical package in order to get the FDA's confidence in approving our product. And if we have strong analytical and preclinical, our clinical work is shorter as well. So anyway, all to say, finding the right people and finding the right teams will help reduce costs in the long run while also ensuring we have the highest quality product, because at the end of the day, this is something going into patients and we're very, very aware that we only get one real shot to make this happen.
And if you can share the details or the way you produce insulin, is that similar to traditional processes out there, or is it a very fundamentally different approach you're choosing?
Again, not the scientist. I think you'll probably have to have a follow-on with Allison, but it's an E. coli-based product, so it's simple in that sense. But there is a reverse phase cleaving step that is technically harder. That is hard to scale, so it won't be hard at the pipette level, but it will be hard once we get to 10 and 100 liters. And so I think that's kind of the big piece that is a little bit more unique for insulin, is that reverse phase cleaving step. And there's a crystallization step, but I can't tell you a whole lot about that. But I know that's another thing that we're thinking about, as far as, can we crystallize it and then ship it to the fill finish site and then uncrystallize it to make it a liquid again. That way we would reduce our shipping costs as well. So anyway, we're thinking about all these different pieces, because at the end of the day, it's about affordability. So there are these unique challenges for insulin reverse phase, and crystallization is kind of like a wish list item, but there are these unique pieces. But overall, it's a pretty standard E. coli based drug development process. So we're doing E. coli, we're not doing yeast or any of the other versions.
You mentioned that the FDA recently changed the regulations, the pathway. So there's definitely a great opportunity there. Perhaps just tell us a bit more about what changed, because I think not everyone listening is familiar with what exactly changed.
Absolutely. And I think this is an important part of our story, and timing is everything. In 2010, there was a FDA rule that had come out that was that biosimilars would essentially receive a fast track status the way that generics do. That status, and I don't remember the official pathway name, but that pathway was introduced in 2010, but kind of rolled out and was projected to roll out for 2020. I started working on this in 2021. When I started, that pathway had already existed, which meant that the math of making biosimilar insulin made a little bit more sense, because it wasn't a full clinical investment would be needed. So that's kind of the FDA pathway. Again, that's very basic version. I surround myself with very good people, so you'd have to ask regulatory about that. But again, on the basic level, that's the gist, is that they made biosimilars similar to generic, and the fact that the path through clinical trials is much shorter. So it would only be a shortened phase 1. And if that data is really good, then you don't actually have to do a phase 2 or a phase 3. You would just have to move to the PPQ batch, which is more about a manufacturing proof than it is about the viability of the drug. It's the viability of the drug at scale.
This new pathway opens exactly new avenues for what you're doing, developing a much cheaper biosimilar, especially in those areas where we have well-documented data and a lot of clinical data.
Exactly.
We've covered a lot of ground with Eric Moyal already, from the roots of the affordability crisis to the realities of building an alternative insulin. And there's still plenty more ahead. In part 2, we keep the conversation going and dig further into how Project Insulin turns its mission into practice. If this resonated with you, please leave a review on Apple Podcasts or your favorite platform. It helps other scientists find the show. Thank you so much for tuning in today, and we'll see you in part 2.
All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.